This presentation is about flow cytometry by Rosa Medellin. Background. The technique of flow cytometry emerged more than 40 years ago. The initial flow cytometers could only measure a few cells at a time. Presently, flow cytometry is a more sophisticated technique to measure the chemical and physical properties of an individual cell. The evolution of the technique allows for the measurement of thousands of cells in a matter of seconds. The advanced contributions of the science field as well as technology allows for the flow cytometry efficiency. It serves as a template to measure cell characteristics useful for quantitative and qualitative analysis. Image 2, a manual flow cytometer with no electronics present. This was the initiator of modern-day flow cytometry system. Principles of technique. The main four components for a flow cytometer are fluidic system, optics, detector, and electronics. Application of technique in general. Fluidic system. The cell sample or a mixture is injected into a stream of sheath fluid with the presence of a saline solution. This sheath pushes the cells into the core of the hydrodynamic focusing region where the cells form a single line. Optics. A cell at a time exits the hydrodynamic focusing region. Multiple laser beams scan the cell, providing a forward scatter light, which is proportional to the surface area of the cell, as well as a side scatter light, which is proportional to the organelles or granules present in the cell. Detection. The cell interactions with the laser lights are detected by photodetectors present inside the flow cytometry system, registering the shape and size of the cell. Each light measures any potential organelles present in the cell based on its fluorescent signal intensity. The fluorescence markers consist of different wavelengths, providing qualitative and quantitative information of the light detected cell. Electronics. The flow cytometer connects to a computer system with a sophisticated software, receives a signal from the photodetector and processes it as an image on a graph where the forward scatter and side scatter are analyzed for further interpretation. Image 3 shows the aligned cells in a hydrodynamic focusing with presence of sheath fluid, and is composed of the sheath fluid hydrodynamic focusing region, and you can observe that the cells are aligned in a single line. Image 4, a schematic overview of the flow cytometry setup. This is inside the flow cytometry apparatus. Improved flow cytometry cross-matching in kidney transplantation. Flow cytometry technique is the most useful sensitive cell-based method for detecting donor-specific antibodies in kidney transplantation. The technique detects all the antibodies bound to donor cells and not only donor-specific antibodies against human leukocyte antigen molecules. The assay measures the extent to which the recipient's antibodies bind to donor lymphocytes. The recipient serum and donor lymphocytes are incubated with fluorochrome conjugated secondary anti-human IgG antibodies. The levels of fluorescence relative to negative control reflects the amount of DSA bound to donor cells without discriminating between specific antigen and the recipient's serum. The importance of using flow cytometer is to evaluate early graft outcomes. One of the drawbacks of the flow cytometry technique is that it is unable to discriminate between complement fixing and non-complement fixing human leukocytes antigens. Figure 1 is represented to your right, and it shows the flow cytometry cross-matching. This idea is based on measuring the binding of donor-specific antibodies to donor lymphocytes when no donor-specific antibodies are present. No binding occurs and the result is negative. When donor-specific antibodies bind to lymphocytes, the antibody can then bind fluorochrome conjugated anti-human IgG antibodies. As you can observe in figure 1, the blue areas on the graph represents the cells mapped by the flow cytometry system. In this figure, The donor-specific antibodies bound to the lymphocytes are represented in the positive cross-match section, the blue area on the graph, by the photoreceptors of the flow cytometer. The result for the positive cross-match is a cell widely spread towards the x-axis. This indicates the cell occupies a large area. Compared to the negative cross-match cell, it is highly marked on the y-axis and not much space is occupied on the x-axis. The cell on the negative cross-match is smaller in size because it is not widely expanded across the x-axis, but the side scatter light reflects the space occupied by its components. The positive cross-match did not map the antibody and donor-specific antibodies separately because they are bound together. Only one image results. If the lymphocyte does not recognize an antibody, then this particular antibody will occupy a separate space in the graph where the presence of antibody and lymphocytes can be distinguished. And this is the flow cytometry is very essential if more cells were in the recipient of the mixture of the serum being tested, and it could identify any foreign cells present. Questions to you. The flow cytometry technique is essential for organ transplantation, providing analysis of donor-specific antibodies interactions with the recipient serum. Do you think flow cytometry technique can be applied to isolate human immunodeficiency virus, HIV, from a patient's blood plasma? Thank you for listening to my presentation, and these are my references.